The cytoplasm of a eukaryotic cell is embedded with several membranous organelles, of which the endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles are referred to as an endomembrane system because their functions are coordinated. The endoplasmic reticulum, or ER, for instance, synthesizes, processes, and transports proteins and lipids to the other organelles, as well as metabolizes carbohydrates in liver cells and detoxifies drugs present in the body. Microscopic studies reveal that the endoplasmic reticulum occurs as a network or reticulum of tiny tubular structures dividing the intracellular space into two distinct compartments. The luminal compartment, which consists of the space within the folds of the endoplasmic reticulum, and the extraluminal compartment, which consists of the cytoplasm that lies outside the folds. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, or RER, and smooth endoplasmic reticulum, or SER. The rough endoplasmic reticulum has ribosomes attached to its surface and is mainly found in cells actively involved in protein synthesis and secretion. They are continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum, on the other hand, is devoid of ribosomes and hence appears smooth. It synthesizes lipids and lipid-like steroidal hormones such as estrogens in the case of animal cells. Did you know that the smooth endoplasmic reticulum present in muscle cells is called the sarcoplasmic reticulum, and that it stores and pumps calcium ions? Apart from the endoplasmic reticulum, the endomembrane system also consists of the Golgi apparatus. Named after Camillo Golgi, it consists of a series of flat, disc-shaped sacs, or cisternae, stacked one above the other. Each cisterna measures 0.5 to 1 micron in diameter, and their number may vary in each Golgi apparatus. The cisternae, concentrically arranged near the nucleus, have two distinct faces: the convex-shaped cis, or the forming face that faces the nuclear envelope, and the concave-shaped trans, or the maturing face that lies opposite the nuclear envelope. Though both the cis face and the trans face are interconnected, they are different. For instance, the cis face has a thin membrane, whereas the membrane of the trans face is thick. In a cell, the Golgi apparatus acts as a packaging and forwarding center. For instance, vesicles containing proteins synthesized by the ribosomes of the rough endoplasmic reticulum get pinched off the surface of the rough endoplasmic reticulum and fuse with the cis face of the Golgi apparatus. Here they are modified in the cisternae before they are released from the trans face. This explains why the Golgi apparatus remains closely associated with the endoplasmic reticulum. In addition to being a packaging and forwarding center, the Golgi apparatus is an important site for the formation of glycoproteins and glycolipids that form a part of the plasma membrane. Lysosomes, another member of the endomembrane system, are single-membrane vesicular structures formed during the packaging process in the Golgi apparatus. They contain hydrolytic enzymes such as lipases, proteases, and carbohydrases that digest lipids, proteins, and carbohydrates, and also nucleic acids. Apart from lysosomes, the endomembrane system also comprises vacuoles. On the lobed inner membrane called tonoplast, a vacuole contains water, sap, excretory products, and other materials not useful for the cell. The tonoplast of vacuoles in plant cells aids in the transportation of a number of ions and other materials against their concentration gradients into the vacuole, which is one of the reasons for the higher concentration of ions in the vacuole. In plant cells, vacuoles can make up 90% of the volume of the cell. These vacuoles also perform different functions. For instance, in amoeba, the contractile vacuole helps the organism excrete waste products and also in osmoregulation. On the other hand, in protists, food vacuoles formed by engulfing food particles provide nutrition to the organism. All the organelles of the endomembrane system work in a coordinated manner and play a vital role in biosynthesis, processing, packaging, transport, and secretion of proteins and lipids, as well as in storage and digestion of wastes.